And connective tissue is what we're going to start with. And as I mentioned in lecture, connective tissue consists of two things. It consists of specialized cells like the fibroblast, chondrocyte, osteocyte, adipocyte, depending upon which tissue we're talking about, surrounded by a matrix. And the matrix is comprised of those protein fibers and ground substance. And I said that the protein fibers, there are three types. We have elastic, which it says are very thin. They provide for stretching and also recoiling of the tissues. Collagen fibers, which are the thickest fibers you see, they are very thick, they're strong, they provide for some stretching, but again, they allow the tissues to bounce right back, which is beneficial, especially in some types of tissues that I'll point out. And then there are reticular fibers, which are kind of the mesh work that kind of provides the structure for the tissue. In this class, I may ask you to identify elastic protein fibers and collagen, but not reticular. Okay, so I could ask you to identify elastic fibers or collagen, but not reticular. And then the ground substance is basically all that filler around the protein fibers, around the cells. And we generally describe the ground substance as being of varying consistency. Solid if it's in bone tissue, semi-solid like with cartilage, or fluid if we're talking about the blood, right, in the plasma. So again, this is what we talked about in lecture. Connective tissue, special cell that secretes a matrix. The matrix consists of proteins and ground substance. All right? Any questions so far? Okay, so four main types of connective tissue that we have. Connective tissue proper, where the matrix will be variable depending on the tissue type. So if we're looking at, for example, a loose connective tissue, proper areolar, for example, you're going to have more liquidy, gelatinous kind of fluid that makes up the matrix. But if you're looking at like a dense connective tissue proper, it's very possible that you're going to have a lot less matrix there, and it's going to be probably a little bit thicker. So it depends on the connective tissue proper. But generally speaking, the fibroblast is the cell that secretes the matrix, with the one exception. Does anybody remember what the one exception was in the connective tissue proper that we talked about this morning? Yes, adipose, which had the adipocyte. That's the one exception.
Well, an areolar is fibroblast, right? Cell type.
Yes, areolar connective tissue is the fibroblast. Dense regular, dense irregular, all the fibroblasts. And again, as we go through this lab, we're going to write this down again. There's space for you to do that. Cartilage. Here, matrix is semi-solid. It's a little bit more flexible. The cells are the chondrocyte, which are found in lacunae. That's plural, lacuna is singular. And then bone, where the matrix, of course, is solid, more solid than cartilage because bone is not flexible. Cells include osteocytes that are found in lacunae. And then we have blood, where the matrix is fluid because it's the plasma. And the cell types that are found there are erythrocytes and leukocytes. Platelets are not actually cells. They are fragments of, does anybody remember the name of the cell? That is the big cell that the platelets, which one? Yeah, it does. And think about large. What's like a prefix for large?
Mega.
Mega? You guys are a good team. Yeah, megakaryocytes. Megakaryocytes, right?
Which one?
For platelets, yeah. So technically, platelets are not cells. Erythrocytes and leukocytes are. And why else is blood special in terms of its cells that are found there? Because the cells don't do what? Well,
Create the matrix?
but they don't create the matrix, right? Like all the other special cells, the fibroblasts, chondrocytes, osteocytes, all of those actually secrete the matrix. But in blood, the erythrocytes and leukocytes, they do not do that. The plasma is actually derived from tissue fluid.
What'd you say about with the ground substance, lesser compact bone and blood, did you say?
Oh, in terms of consistency?
Like right here for these?
So it'd be solid. No, we didn't go over that.
Oh.
We're gonna go into page, actually page 2, if you look, there's something there about muscle. We're gonna get to that a little bit later, and nervous tissue. So there's some information there about these tissues. But let's go ahead on to page 3 and look at the connective tissue. We're gonna start out with connective tissue proper. And the cell type, do you remember what it was for areolar connective tissue?
Fibroblasts.
It's the fibroblast. The function of it is to fill space and provide flexibility. And you do find it around blood vessels and nerves. Areolar connective tissue is like the bubble wrap of the connective tissue world. You're gonna see it everywhere. In fact, if you guys did a cat dissection at any point, I don't know if you did last semester or if you ever did one, but when you open up the cat, you see that there's sort of this spider-like tissue that's in there. That's actually areolar connective tissue.
So are we gonna do that instead of looking into numbers?
No. No, that was last semester. But you do have in your slide box, you should have areolar connective tissue. And you can take a look at it. And a lot of you guys are gonna have, I think you have like a purple stain. Mine kind of looks like the picture I showed you in lecture. So I wanna show you what mine looks like. Again, yours is probably gonna be purple stained. I think most of them are just purple. So, yes, they have the need to teach you with the stick. Alright, so over here, I wanna show you a couple of things. First of all, when you see these,
Areolar, right?
yeah, areolar connective tissue. You see the dark purple circles there? Remember what those are?
Fibroblasts?
Those are fibroblasts, right? You can see those. And then if you have any thick, like the thick pink fibers that you can see on here are what, do you think?
Elastins or what, no?
Okay, remember there are 2 types of protein fiber you have to identify. Collagen and elastic, not reticular, because you have to finely adjust and sometimes it's tricky on the eye. I'm not gonna have you identify it. But the thicker salmon pink colored fibers that you see there, that's your collagen. And then the dark purple fibers are the elastic fibers. And what's cool, if you see over here, you've got one that's kind of broken. Notice how it like kind of snaps and coils like very, very tightly. Can you see that? Because these fibers are so elastic, you can see that when they break, they show that elasticity. They try to get, into a small position. So go ahead and take a look at yours and see what you think. Again, you might have one that's just all dark purple stain. Mine is mixed. My stain is. Honestly, I'm probably doing this because this sticky stuff is great. The more time you take it down, the harder it is. Exactly. But then those silly frames, the cardboard worked because it was humid, and I ended up just using all the frames for pointers. And that didn't go so well either. You might be in a precarious spot. This seems like it was holding pretty well, but it may fall down. Sue. Anybody have any questions about it? What you're looking at?
Just wondering on what is, are you gonna show the things in purple on the side, so?
Yeah, I'm gonna probably show you this slide. So it'll be like the pink fibers or collagen, the dark purple or elastic.
Alright. Cause on my slide it shows pink and purple, actually it's different than that.
What do you mean?
I mean, it shows like the way you're describing it, showing it, pink and purple, you know?
I don't know. Oh. Oh, yeah, yours looks good.
Yeah, cause on yours it's kinda like orange.
Right. Yeah, but still your wider fibers that are more like pink, those are collagen and the thin ones are the elastic fibers. And then
Okay.
you got a lot of fibroblasts in that. Everybody okay here?
I am, yeah.
Cool, see, it gets easier.
I don't know, it's fine.
The only thing that you have, you have one that's all purple. I'll get you. Yeah, that makes it a little different. I understand why you're confused. Because this is a little confusing. Doesn't look like mine. You don't really seem to have a whole lot of fibroblasts, but if you look at the tip of my arrow, you see there are thicker fibers there. Here, let me see. Yeah, like see the thicker fibers are lighter purple. That's your collagen. And then all the dark purple in the arrow. Huh? Yeah. And then like all the dark purple little fibers that are around it, that would be your elastic stuff. But I don't see a lot of nuclei in there. There's not a lot of like little, I don't see any circles really. Yours is a little bit different. Okay, anyone else not sure what's what? Okay. What? Did someone have a question? You did? No. You're welcome. I need to wear glasses. I can't see who's looking at me or not. Okay. Alright, so next we're gonna look at adipose. Of connective tissue proper, this is the anomalous one because the cell type is not the fibroblast, it's the adipocyte. Its job is for protection, insulation, energy storage.
What cell type is it?
Adipocyte. Or adipocyte, yeah. Now for this, you do have adipose tissue in your box. A lot of you are gonna have the slides that are stained dark purple, which I don't think are as good because you can't really see as much. Some of them are gonna be not stained. You're gonna be able to see the lipid droplets. Mine is not stained. If you have one that's really pale, remember to adjust your light level low because otherwise it'll appear washed out. You won't be able to find the sample. So do that. I'm gonna show you another place where you can find adipose as well, but we can look at this slide first. That's what you're gonna see if you have one that's not stained like me. This is a big vacuous adipocyte, and its whole job is basically just to store fat. Sometimes you can look and some of them get smashed, you'll see like a little lipid droplet coming out. And every so often you can see a nucleus, but I don't really see any on mine. Mine are just here. All along here. Fat stored in there. You can see there's not much matrix at all, if any.
Ours look like just purple
Yeah, yours, you have, I think, the niggery stain. I'll show you another place where you can find it that's far more interesting. Do you guys remember which slide is the skin slide? Which epithelium that we looked at last week? Yep, the stratified squamous epithelium that's keratinized. If you go back to that slide, which we're gonna do a couple times today, if you go back to that slide and you look at the hypodermis, right, the tissue that's underneath the dermal layer. Last week we looked at the epidermis, so that's the stratified squamous epithelium, it's keratinized. If you scroll down and you get into the hypodermis, which I'm gonna show you, you'll also find some nice adipose, and it's gonna look a little bit more interesting, I think. This is why I like to go back to this slide. Don't forget too, some of you guys have to flip your slide over because it's upside down. Remember if it has an arrow. Alright, I wanna show you, this is what you looked at last week.
What was this?
This is the stratified squamous epithelium that is keratinized. So this is your skin, okay, this is your skin slide. And everything you're looking at up here is the epithelium we saw last week. Down here is the dermis, and if we keep scrolling down, 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 now you're gonna see pockets of fat. See, this is the hypodermis. So all of this is adipose tissue. And again, it's a lot more interesting looking, I think, on this particular slide. And when you zoom in, you can see more of the features of the cells. You'd be able to even maybe see a nucleus or two.
So for the test, can't you just fill that slide in?
Yeah, more than likely I will, but it's nice to see.
I have
A little one?
I don't know. I think I see it, yeah. They look like little bubbles instead of big bubbles.
Yeah, like basically something like this.
Yeah.
Yes.
A lot of them.
Great, because nucleus is like over here. Because the cells are just full of fat, the nucleus gets pushed out to the cells. Yeah, how did you get lucky?
Oh, yeah, you
You
just.
can ask us that about the nucleus on there.
No one asked that.
Big fat bubbles.
I'll ask, you know, what's the function of it, or you might find
I
it.
think sometimes the slide just sucks.
Yeah.
Travel subject,
I use stuff like all the time.
That's why I get a good one usually. It's not,
I think those are air bubbles.
I don't even know why I'm doing this if that's going up there.
It does.
Do you know what I mean? Like,
I feel like I went there. Okay.
We did have one, I saw.
here's a stupid question. If you're putting the slide on the TV screen, this is gonna sound ignorant, I apologize now. Why are we looking at it in the microscope? Because it's so much easier to identify it on the TV screen.
Yeah, that's ignorant. No, I'm joking.
With all due respect, I feel like this is an unnecessary process of learning.
No, the idea for you is to get familiar with seeing it, the characteristics of the tissues for yourself, so that when I put it on a TV screen, it will be easier for you. Like, again, it's more active learning on your part if you're actually looking for the tissue and you're looking at it and observing characteristics for yourself.
No, I understand, I'm just. it's
No, I
frustrating
understand your
because,
question
like,
too.
most of
And it
our
wasn't-
specimens or slides are not.
And it actually wasn't an ignorant question, it was a good question. It's a good question.
I wanted to preface, like, I wasn't just running my mouth, like I.
Sure, no, I got you.
Would be good if we come in and turn TV on. Would be good if we could come in and turn TV on.
Well, technically you could, I mean, there's no lock there, thank you. I knew I forgot something over there, I just didn't know what yet. Your question threw me. Did you want,
No, I took it off because it made me mad.
It made you mad?
I couldn't see, I mean, the detail that you have on there is so much more than I could get, so.
Did you look at your skin slide?
No,
Look at that, cause then you'll be able to see what I'm looking at it there. I think if you look, let's see here, so you wanna get your keratinized one. Here. Get that on there, and you go to the hypodermis part. The part that's real deep. Here's, you might have to flip over.
But it's on the stratified squamous up the other slide.
Not that way, the other way. In other words, like upside down, yeah, hold on. I think,
Is it stratified?
No,
that's been
yeah,
my problem
that's,
the whole time.
she's showing the one from the stratified squamous slide.
no, I don't think so, because most of the labels are on right. It's just that one for some reason. For a lot of these companies, somebody put them on backwards.
You said it says stratified squamous. Okay.
We're supposed to look at the stratified squamous straight.
The stratified squamous keratinized, look at the very bottom, and the hypodermis, that's where the fat is. Here, you want me to look?
Should I check mine and make sure?
Okay, yeah, okay, you know what, turns out we were right. This one didn't have an upside down label. So, we'll get this so it looks right. Okay, so you got a lot of nice pockets of fat here. Down
Just
on
say that to all the students.
Yeah, it is funny, the things that you say that are part of the job, like if somebody else heard it, it's like, what? Yeah, like where the arrow is, if you zoom in on that, maybe adjust the light a little, you get some good contrast. You'll be able to see what you see on the TV screen.
Thank you.
Yeah, you found it. Now you can make the light a little greater so that you can see a little better. You found it.
I was be tweaking, so no.
That's okay, if you want it look like real low, as long as you can see some of the detail. It's down, yeah, but now yours, right, but yours I think is not the one backwards. Thank God.
Yeah.
This is not very nice. Which isn't the skin slide, but you still have some real nice pockets here. No, you're fine, you can make difference. Anyone else not sure?
See that Apple Watch?
Mm.
See that Apple Watch?
Basic.
It's fake.
What?
It's fake.
It's fake? How do you get a fake Apple Watch?
Market district.
It's real.
Not thinking that you're.
You
Go to
turn,
China-
you
go
turn
to
it on,
Chinatown,
it's a
New York.
pear instead of an apple.
Yeah.
That's a big one.
Yeah, I don't see any adipose stuff. I don't see any of that.
Go to Chinatown in New York and get a fake Apple Watch.
That's what mine looks like. Yours is like that? Mine looks
Doesn't it
like.
just look like white circles?
Oh, you're on the wrong side. I'm on the wrong side. Yeah. Like that.
You gotta turn yours upside down. There's an arrow on it.
Oh, yeah, that too. But
This one,
there's an arrow.
There's an arrow.
this is
To
your
turn,
line.
so you flip it the other way because it's upside down.
Wait, the blue arrow? What are you doing?
In the top left corner.
How did they adjust? It adjust down here, and then that moves it up and down. But I don't see what.
Flip it.
Oh really? And it's
Yeah.
a whole different.
No,
thing.
not that way. Like this way.
I did the same thing, man. Don't feel bad.
It's like,
Now
make
it's
sure you look at it.
epidermis, and then you go down.
Where is that?
Quite as
Adipose
like,
tissue.
I'm trying to where's that at? What layer?
Below
How
the
important
epidermis.
person
You gotta go like find this stratified squamous epithelium and then go down.
You can see,
So you
you
see
can
like
see a
that.
cell there.
I'm about to flip the whole thing.
Where the arrow is?
Yeah.
Right on his
Turn
head.
it around.
Right on his head.
Okay, who's still not sure? Did you get a good one? Oh, cause I was gonna say, if you get a good one, she can look at it, and it'll be better. Yes.
Using my brain roots.
Yeah, that's good. Looks great. Anybody else?
Yeah, I think we got it.
Okay. Alright. Okay, so if, done with that one, then we can move into some other types of connective tissue proper. These are the dense connective tissue proper we talked about today. And these are not in your slide box. Dense regular connective tissue. This one you have to look at on the poster, you have to look at it. But I had a nice picture of it for you in lecture. So this is dense regular connective tissue. You almost can't see anything but the collagen fibers. And there are some fibroblasts that are kind of in between the collagen fibers, but they're kind of hard to make out. Where do you find dense regular connective tissue? Good. Remember, because the collagen fibers, they're all arranged parallel to each other. So you can pull on something in one direction, it bounces back in the same direction. The tendons and ligaments are kind of there. And then, again, the special cell is the fibroblast. And now we can move to dense irregular connective tissue. And if you have your skin slide, the one we were just looking at for the adipose, if you have your stratified squamous epithelium that is keratinized, if you look in the dermal layer, right here, you just move up from where you were in the hypodermis. Here, I'm gonna look out so you can see. If you move up into the dermis, that's what all of this is the collagen. So above that, we've got our epidermis, but all of this area here, this is all dense, irregular connective tissue. Now, it looks weird. It's a weird looking tissue. All this dark purple stuff is collagen. And remember, as we said in lecture, that as we get older, the fibroblasts that are found there don't tend to make as much collagen, which is why the dermis gets thinner and skin gets thinner, and it isn't as elastic. It doesn't bounce back as easily. The reason why you see all this collagen in weird ways is because some of this is a horizontal, like you're looking at a horizontal view. Some of it might be diagonally coming at you. Some of it might be like a cross section, because the fibers are oriented in all different types of ways. And so that's why the tissue looks so weird. But all that dark purple stuff, that's all collagen that you see there. And there are some blood vessels that are found there as well. You can see some of these big circular areas. That's what that is. So anyway, go ahead and take a look at that. And if you're not sure, I'll come around.
My slide is not okay.
I was just seeing if you got on camera.
Yeah, I'm pretty sure I did.
No, you did. You're good.
My whole
I
face.
zoomed in real tight to the TV screen.
Thanks.
Now, audio-wise, you're gonna be like, right? So that's okay though. I mean, I'm gonna put it on YouTube, so
What's your YouTube channel?
I'll send everyone a link.
You're in this video.
No, I mean, just your voices, because I'm zoomed in real tight.
Oh.
I like that, that's very, I saw that earlier today, felt that was in the studio. It's official.
What,
What is the
what's,
I get pretty nerdy.
what's your name? I didn't look
Oh,
at
it's
it.
Mike.
Just Mike.
That's my name.
Go to what
You know what I'm saying?
he
What's your-
posted it
what's your
before.
YouTube?
Yeah, I'll put a link, but after they're all
I just want to know
online,
what your name is
I don't
on
remember.
YouTube.
You posted a
Nothing
video
fancy.
for anatomy.
Yeah,
Yeah.
it's like probably my last name, some derivative of my last name.
Yeah, all of that's
Like,
college.
I think it's like M Fleet.
How many subscribers you got?
Maybe 6.
I'll subscribe to one.
Make sure you click the bell for
up, guys?
notifications.
This,
It's your boy.
Fuck yeah, I would watch it.
called,
It'd be better if you recorded yourself through the whole thing, not her.
Yeah.
Or she, like, it's just a selfie view for 3 hours of you taking
Like
videos.
picking my nose.
Yeah, where your arrow is, that's all.
How mad would you be if you're like, man, I really need that lecture? Oh, you looked me up.
Just sit there and watch.
Yes.
Like, you can't really even hear the lecture.
I know.
Yeah, it's just your heavy breathing.
She says something like,
I mean, I might have, like, written part of the test, I might ask questions about the stuff that's not inside box, but I'm not gonna have it. That's one thing. You'll never find it in that stuff. It's not in the box, so.
What's the subtype for the dental?
It's fibroblasts, yep. Yeah, so you got loose and you have dense. What makes it dense is the collagen, and that's, you know, dense regular, and then this is dense irregular. But the cell type's the same, fibroblasts.
How'd you get into this? How you changed it, it looks like that?
I zoomed up to dermis and I magnified it.
Oh, you just kept scrolling on.
Yes, I found where the dermis was and then I magnified that area.
where's the dance room?
All of that, everything that's on there. It's all dense regular, irregular connective tissue.
Oh, it's your
Yes,
I thought that was yours.
Yeah, dense
We're not
irregular.
going-
Irregular.
irregular. We're not.
Oh, it's mixed in both.
Dense regular, we
It's
don't
on
have in the slide box.
No, I
just
have
showed
it.
it to you on the post. Dense irregular is what you're looking at all up on the screen.
Look at it,
So
super
weird,
regular.
like it's way.
It is a weird looking tissue.
This one is this camera.
Is
Something like
that a dark purple on that thing?
I think she said collagen is a dark purple.
What's the light purple?
And I kept looking at it on here
I can't tell the difference.
I thought the light purple was.
and I started
Maybe
at
I'm wrong.
Lois and it
I got
was
red.
the sauce.
Yeah.
Yep.
I feel like I'm looking at a different.
That's- that is not- Yeah, you're right. You
Background stuff
might
is
have an
called-
alien brain.
The background stuff is called the ground substance or the
Oh, yeah, this is
matrix.
different.
Ground substance.
I wonder why I did so bad on the quiz.
Blame it on that.
Yeah, that's what I'm gonna blame it on.
This is something
I
like
didn't didn't study.
That one has an arrow,
Yeah.
so I don't think this is the other way. Yeah.
Ooh.
Well, not that way. What you wanna do
This is the first one we did.
like this is the laser.
Oh, okay.
I was
I
too
don't
busy
think this
figuring
box
out the
sounds much
microphone.
bigger than this.
So fibroblasts for regular.
Is that what you're
No one sits there, so it should be okay. But yeah, in time, somebody else will have this like.
Okay, beautiful. Just not us.
Yeah, you got a good box now. Alright.
Thank you. I need some more stuff. I fucked it.
Oh no. No, we got a good one. You good? Good.
What are the super dark lines? Is that the collagen?
Yeah, the dark, the real light lines there.
Highly colored.
Save me.
Okay.
I'm like 90% sure.
So like yeah, all this, this is collagen here. Sometimes you see it at different angles.
Uh-huh.
So it's gonna be sometimes like little cross sections, sometimes it's horizontal,
Okay.
sometimes it comes out at you more like on a bias.
And what's the lighter stuff then?
All this?
Yeah. No, the in between the dark and the light. You know what I'm talking? It's like the light thing.
Matrix.
Just the
The
okay.
ground substance.
Okay, that's what I thought.
Yeah, but I mean, it's dense because all the collagen, but you do still have some space there, you still have ground substance.
Okay.
And then the two openings of the blood vessels.
Yes, these would be like a blood vessel, probably the small arterioles or something like that. Those are pretty open, like that's probably a venule because it's collapsed. The wall's pretty thick, so it's probably just a smushed arteriole. I'd guess. Anybody else? Anyone else?
You just said we're doing bone charts.
Nerves.
We're doing what?
Did you say we're doing bones, Jen?
Yes.
And that's,
I'm gonna try to finish this if I can.
Okay.
Well, we're just gonna go as far as we can. Yeah, I like to
What
finish,
are you talking
definitely the connective tissue, the
Skin,
muscles.
right?
Maybe if we have nervous
Yeah,
at the end of
it's
that.
around like
But I wanna go so
blood
fast,
vessels and
but this is
stuff.
too soon. Oh, and stay here, this is here, that's why we just go to the Jones.
Yeah, capsules and sheath.
I like it though, it's a big bottle, it holds a lot of water, I always leave
I'll see what I can do.
it. Yeah, well, you use it though.
Yeah, put the sheath in the dermis, whatever that means. Put the sheath in the dermis. And then, yeah.
I actually missed that. And when she came back, she was always like, okay, come on. Cause she did my work, like get like, you know, stuff alphabetized and stapled and all that. And then she's like, come on, gotta go to the gym now. I'm like, she was the best lab assistant in the world. I got nap. We have 12-hour days and take a break. We had all that down. We did, we went to the gym recently. I'm not even in the office. She was really funny. I really do miss having her on. And the students liked her too. She was a hoot. You would have had so much fun in class when she was here. My old
Who says
class.
we're not having fun?
Huh?
Said who says we're not having
Well,
fun?
I don't know, you're asking ignorant questions now. No, it's just, no, this girl was hilarious because she was like, I was always the straight man trying to be professional, but she would always come in and like, she would always say something, and a lot of times it was like one of those things that was completely juxtaposed to the way I was trying to be. It would always be something ghetto. It would always be something, you know, like really funny that you wouldn't see coming, but then it would kind of be in a way, it was sort of a slap at me. She's the one who bought me that t-shirt. I'm glad I didn't get to PH today. I'm gonna have to wear that next time I have a second chance. It's really funny, because she would always have that. She was great.
sounds like a party, bring her in for me.
Well, she just graduated from Pitt. She has her master's now. She's getting a job. She's getting a real job.
Yeah.
That was a while ago, she was here.
Yeah, she was my lab assistant. Well, one day she just came in. I had a lab assistant. She's like, I work here now. I said, what? What do you mean you work here now? She wanted to work for me. I don't know why. And my other lab assistant, she ended up working for Kevin, I think. I mean, I don't know how. She just came in. That was it. Yeah, she was funny. But I think she was my lab assistant from 2016 to like 2017, I think, through that time. Yeah, it's funny. I really do wish you could have met her though.
What slide are we on next?
I don't know. I was thinking about the time that she photoshopped my face. One time she photoshopped my face and then I didn't know what a tramp stamp was. So she photoshopped my face. I have to show you.
Did she make you a tramp stamp?
No,
Oh.
she actually, no, she had my face. There was a picture she had, and there was some man, right? A picture of a man erasing a chalkboard, and you could see a tramp stamp there. And she turned my head, like, I mean, it looked like I was like this, you know, I mean, it's a natural. I have a picture. I'll show it to you guys. You'll like it. Yeah, but those are the kinds of things she used to do. Just kept you rolling.
Sounds like it.
I think you're gonna be the best teacher I ever had.
Well, I'll be the most
See, it's your question. I just can't.
Alright, so the next slide, Lagaya, you wish to ask, are we on it?
When I get back.
Yes, ma'am. Okay, here we go.
Eileen Carter.
So next, we're gonna go on to talk about a couple of things that you don't need to actually identify, but you, again, just like with the epithelial tissues, you have to be able to talk about, like, you know, where you would find it. All the information on here that's on this handout about function, the cell type, and all of that, you should know that for all of these tissues, but only the ones in your slide box do you have to identify, just like with the epithelial tissues. So these next ones, they are not in your slide box, but they're up on the poster, so we'll talk a little bit about them. These are connective tissue proper. So these are also going to have the fibroblasts as the cell type. Elastic tissue, we have right here. You can see a lot of elastic protein fibers in the matrix. So some of you guys, you remember, we looked at areolar connective tissue, you could see these stained dark purple. This kind of tissue you're going to find especially in the elastic arteries. So the elastic blood vessels, for example, the aorta, right? So the aorta comes off the left ventricle. When the left ventricle pumps, the aorta has to be able to stretch. And when the left ventricle relaxes, the aorta has to be able to recoil to propel the blood forward, right? So it's going to have a lot of elastic tissue in the wall of it. Again, the cell type is the fibroblast. Reticular connective tissue proper has a lot of reticular fibers in the connective tissue matrix. And you're going to find this in around a lot of organs and things, providing structure and basically a framework for them. You also find, if you take a look in your basement membranes of some of the epithelial tissues we looked at, you'll also find reticular fiber there. Reticular tissue there. Any questions about the connective tissue proper?
You're not gonna ask cell type function for those, it's around the slide box,
No, I could. You should know, for all of the slides that are listed in this handout, you should know cell type, you should know, like, all the stuff that's written, the function, those types of things, but only you have to identify the ones that are in the slide box.
Okay, it's like elastic tissue, I thought you said identify that. I thought you said that was one you had.
No, it's not in the slide box. Okay, so moving on to cartilage. 3 types of cartilage. What's the cell type? Does anybody remember?
Tense.
Oh,
Sorry.
Starts with a C. Chondrocytes, right? And they're found in little chambers called lacunae, right? And so the one that you do have in your slide box is hyaline cartilage. The other 2 you don't have, so let's talk about them first, and then we'll take a look at hyaline cartilage. So fibrocartilage, the function is to support things, of course, and provide some flexibility, and you find it in the intervertebral discs. Basically, the fibrocartilage, again, if you take a look, the only difference between the different cartilages is the protein fibers that you find in the matrix. Fibrocartilage has a lot of these, you see on this stage, there's a lot of blue collagen fibers there, so it makes it a really tough tissue. It can take the compression forces in between the vertebrae, so that's what makes it good in the vertebral column. Elastic cartilage over here has a lot of purple elastic fibers here in the matrix, and so it's really good to have in places where you need to be able to move something, like the epiglottis, for example, right? That's going to be elastic cartilage, or like I said, the pinna, the outside of the ear, right? It provides support, but if you bend it down and you let go, it bounces back because there's a lot of elastic fibers there. So those two, again, they're not in your box, your slide box, so you don't have to identify them, but you should know the cell type, you should know the function of them. And so the cell type, again, chondrocyte, ground substance is gonna be semi-solid because it's cartilage. Cartilage isn't as solid as bone is. The last one, which we do have in the slide box, is hyaline cartilage. Hyaline cartilage, you're gonna find in fetal skeletons and in fontanelles. And you do have, in your slide box, you should have a hyaline cartilage slide, but I personally like to use my pseudostratified ciliated columnar epithelium slide because this is the trachea. And you're going to see the hyaline cartilage also in this slide very nicely. I think the hyaline cartilage slide, I think for most people is just the cartilage. But this pseudostratified ciliated columnar epithelium slide, you can definitely see a lot of hyaline cartilage there. So again, you've got a glassy looking matrix, it's sort of shiny. I think it's the prettiest staining tissue that I have. All of the chondrocytes, these little round circles, these are the chondrocytes, and the white space around them is the lacuna. And then all of this salmon pink color stained area is sort of shiny. That is the matrix, which is glassy, and therefore it's called hyaline cartilage. Yep.
I'll get it on here.
Oh goodness, no. I think, oh, you know what, you did this again. See this thing right here? That's what you're looking at. If you need to adjust the light down, you got, there's a little lever somewhere here. There's the lever. There is a lever someplace. Sure. Ah, here. See this
This
right
looks
here,
like
this little screw? That's how you adjust the light. Well, wherever is best for your eyes. You're in the right place. You just have to zoom in on it and maybe adjust the contrast a little by using that lever, but that's what you have.
Yes, I am.
Yep. Looks good. Yeah. That looks very nice. Yeah, that looks nice. You do got it. Anyone else have any questions? Sure. You're okay. Yeah, we can always get like a different, yeah, that's definitely. Yeah, we can get you different blocks if you have, you know.
Did the change and then the second one, like not some good spots too.
It's not your day, you and I are having the same kind of time period in our lives. I like that one, I think that looks pretty.
You can look at mine, it's pretty good.
You got it, okay? You got it, okay, then, you're good? Yeah. Okay. of connective tissue that we're gonna look at is bone. And then finally, we'll do blood, and we'll finish up with connective tissue on that. So, bone is a connective tissue which basically has two forms. You have compact bone. Where do you find compact bone? Well, sure, if you're off the end, but yeah, outside of the bone, you're gonna find it. So if you're looking at the skull bone and you break it, you're looking at the top part or the bottom, it's nice and smooth, that's compact bone. Inside, just like the filling of an Oreo cookie, we have spongy bone, right? So compact bone is the one that we're gonna look at today. Spongy bone, the job of spongy bone is basically to store red bone marrow, which produces red and white blood cells. But not all bones have red bone marrow, right? Which bones have red bone marrow in them?
Spongy.
The spongy bone. Which ones have which bones have spongy bone that contains red bone marrow?
Long bones. Compact.
Not the long bones. Actually, the long bones in adults generally have yellow bone marrow, right? That's what they use to flavor stews sometimes. They use the yellow marrow out of the cow's femur, you know, and that's like, and what it is really a bunch of fat. The red bone marrow has special tissue in it that allows us to make red and white blood cells. And in an adult, and even in young children past a certain age, you're only going to have red bone marrow in certain skull bones, the sternum, the ribs, and the pelvis. Those are the main places where you're gonna find it. That's why whenever they're looking at doing a bone marrow transplant, a lot of times they go in and they'll have to puncture the bone, and they go into the os coxa bone to extract some of it, and it's a very painful procedure for people. So that's where you'll find spongy bone. Compact bone you can find pretty much everywhere outside the bone. So it contains diversion canals, or the central canal, and each central canal usually has one artery, one vein, and one nerve that passes through it, and little canals, which are called canaliculi, through which nutrition is sent from the central canal to the osteocytes that are found in lacunae. So remember that the cell type here that actually makes the bony matrix that builds the bone is the what?
Osteocyte.
Well, the osteoblast builds the bone. But once the matrix surrounds the osteoblast and it gets trapped in there, it becomes an osteocyte. It's an inactive osteoblast is what it is. Okay, so once it's in there, that's what happens. So the cell type, osteoblast, osteocyte, the ground substance is solid, and of course it's there to provide support. I know some of the people in anatomy that were with me last fall, you guys saw this. I was reviewing this textbook that I might use for my pathophys class. Oh my goodness, the pictures they had in it were disgusting. They showed this artist's depiction of what it would look like if you didn't have a skeleton. It was like basically a puddle of flesh. I don't even know how they had like the head up, because I mean if there were no skull bones that wouldn't even be there. You'd probably just have like some eyeballs hanging out. It was so gross.
Like an octopus.
Not even. It was even worse than that. You remember that picture? No, I almost wanted to get the book just for the pictures, but I couldn't use it for class. But I bet you the students would read it. Just for the pictures. Pictures were really gross. They showed like fractures with blood squirting all over the place because of trying to show there's blood vessels in the bone. Gross. I don't know. Anyway, alright, so take a look at the compact bone tissue. You have that in there. And it looks like a forest where the trees got cut down. Or in my case, it looks like the face of a teddy bear.
Yeah, mine's all broken.
Sometimes you gotta find like a good place on there. Mine looks like,
Osteo. Isn't that what the ring is?
Mine looks like, mine looks like the face of this. Actually, this looks like a dog, doesn't it? This looks like,
This is a dog.
a lab, here's the nose,
I see a beagle.
or a beagle.
Spongy bone.
Compact. Spongy bone, actually, if we look at the,
You can't even see mine.
if you look at this, you don't have spongy bone in your mouth.
Mine, the whole thing's cracked.
Spongy bone looks a lot more diffuse, okay? It has this trabeculae, but spongy bone doesn't have osteons. That's the circular structures that you see on the TV up there. It doesn't have that. Compact bone has this. This is an osteon. And again, the osteon has in it, there are many osteons in compact bone tissue. We have a central canal through which the artery, vein, and nerve go. We have osteocytes, and we have these little canals called canaliculi. And that's what that word literally means, is little canal.
We have to know all that again.
If it's on the handout, you should know it. Here's the teddy bear. That's the teddy. I think that's what.
And this is compact.
Compact. Because you see all those little rings, those are osteons.
So basically no compact, no left, no like what it contains.
So much.
Cell type function from ground substance.
What's the ground substance?
Solid. So here you can see that this is the central canal of the osteon, probably a blood vessel or nerve here. The dark structures that you see, these are the trapped osteocytes in the lacunae. It's not as clear as what we saw in cartilage. There's no space that you can see. But these radiating dark structures that radiate from the central canal out into the matrix, these are the canaliculi, which bring nutrition from the blood vessels in the central canal through the bony matrix into the cells that are found in the lacunae. So all that's listed on the handout. You know, those structures, they're listed on there.
So canal is the line was dark.
The what? The central canal.
Now they don't have dark structures.
Yeah, the dark little lines that radiate out like the rays of the sun.
Okay, and the dark circles osteocytes.
In the lacuna, yeah. The dark little spots there.
What's this other part on here?
Let's see.
To the, looks like snake skin.
Oh, that's a crack.
Yeah, that's all mine was.
Yeah, I have that big one.
Yeah, that looks good there. You need to stay here. You need to stay in that little square. I think you somehow lost your way, got off.
Again.
I'm
good.
Oh, you
Looking
can't
at
make out what
I'm
you're
gonna
doing.
need some blood.
So much
It's just,
information
it's
between the lecture and this, like.
It's not easy to make out.
That's the whole thing.
You would ask if you know what it was.
I could say, what are the chambers called? I could ask that. And guys, for this tissue, I could ask you, what's the chamber called that these cells are in? Cause, it's listed in the handout at the beginning. You mean like a low magnification?
Yeah, that's why I asked.
Is she good?
I'm so confused, mine look different, but now it looks, okay.
Yeah, that's what I said. Sometimes it can get tricky if the magnification level is different. And that's why I tell you guys to come study, because the magnification level can make a difference sometimes.
I don't have any of the white circles, they're all black. Yeah, mine are too.
Well, that's possible, again, it's an artifact in the preparation. That's where, still the central canal. Maybe your artery, vein, and nerve are still in there, but this, it might not be there. No.
Add a blue line.
Blue line? Oh, yeah, that's cool. Looks like somebody wrote on that. I can't see them. That's cool. How about that? What's the outer? The outer?
From the last one, the
You mean the rings? Those are lamellae, but that's not one of the words that's listed. Yeah, it's just, the whole thing itself is called an osteon, that whole round structure with the rings and the central canal. That's an osteon. But, and then each osteon has a central canal, and you've got the osteocytes that are trapped in the lacunae in there. And the rings, you have them because of how the osteocytes secrete the matrix. So it makes it, like, you know, like rings of a tree. You cut down, it's the same. Okay. So I guess the last connective tissue we're gonna look at is blood. This is one that I have to say no one has ever missed unless they overthought something and thought it was supposed to be a harder question. Because in terms of identification, blood has to be the easiest tissue to identify, but it's not always the easy tissue to focus on because red blood cells are so teeny tiny, and when you look at this slide, initially, you are going to see a bunch of just pink stuff. Like it's just gonna appear pink. You're not gonna be able to really see any cells until you zoom in. This is what it's going to look like under low magnification. See how it's just pink? But if you get to this point, that's good. Because like I said, it's not easy sometimes to focus on this. So when you get closer though, and even once you zoom in, it's still not like super duper magnified. You really need to look under oil immersion, which today isn't really that necessary because we're not identifying the individual white blood cells yet. We're gonna do that maybe next week. But if you wanna really see, like for example here, this with power magnification, it's 3,100 times magnification. So these are a lot bigger. And like I said, what we can do on these light microscopes is 1,000 times magnification. But this is nice to have. This particular slide is stained with something called Wright stain, W-R-I-G-H-T. And there are a couple of different dyes. There's an acidic dye and a basic dye. There's an acidic dye called eosin. And essentially that's why you have different colors. You have the red blood cells, which tend to attract the red eosin dye. And then the nucleus tends to attract the basic blue dye. And then there are some parts to the cell that might attract both, so they appear purple, and so forth. But today, for our purposes, the cell type here are the erythrocytes and the leukocytes. And we're not gonna learn the specific white blood cells yet, but I'm gonna show you generally what's on this slide. So all of these red circles you see, these are red blood cells. There are many more red blood cells than there are white blood cells. But the larger cells that have the purple nucleus, these are white blood cells that you can see here. And of course, we mentioned platelets that aren't really true cells, but you can see them. They're these little tiny specks that are found in the plasma, which is all the white on this slide. So you have your red blood cells, you have some white blood cells, and then you have these little specks, which are platelets, and the white is all the plasma. And something kind of cool, when you look at the red blood cells, you'll see sometimes there's a white spot in the middle. Because of the shape of the red blood cell, it's concave on the top and concave on the bottom. So sometimes the dye doesn't get in there. And so it's just white in color. So that kind of is evidence for you that they're like that. But that's blood. Right, so the red blood cells, there are many millions of a lot more red blood cells than there are white blood cells. In whole blood, about 45% is red blood cells, less than 1% are
What's
the
the
white blood
ground
cells.
substance for this one?
Huh?
What's the ground substance for this one?
Liquid, it's fluid, it's plasma, yeah. Anything else for this one?
Kinda getting tired.
I know, this one's a little bit longer. Let's just do, well, let's, but then we have to do it on, I don't know if you're
You
gonna.
can always cut things out,
I can't help it that there are 4 different types of tissues though. I can't like.
or we can just go over it and not be tested on it, like, there's an answer for every.
We did that though, remember last week, we had the microscope, I didn't ask you any questions about the microscope, we just went over it.
The base, the eyeballs, the light,
Okay, maybe we should be tested on it. That is not an eyeball. A microscope does not have eyeballs.
But you need eyeballs to use the microscope.
That's funny, no,
what were the little spots in the plasma?
the platelets. You guys are killing me.
And the white one is the plasma.
Yes,
Yeah.
the white is the plasma. Yeah, can't we just go over the muscle? We'll do the 3 muscle, and then we'll leave nervous for next time. And you don't have to, we're not taking a quiz on Thursday, so we have some time. But this way, at least we get through those, and then we just have one little tissue.
So basically, we're gonna have more time to bring more information in, right?
Let's just do it instead of talking about doing it. Let's just do it.
Yeah, I
Let's
think,
get it done.
I think it's a good idea, because we don't have to keep
And get her
You
done.
only have 1 slide, you got the muscle slide, right here. 3 muscles. I'll tell you what, I will show you what they look like, see if you can tell me what they are. If you can tell me what they are, then I will say you don't have to look at them, because you need to see, how's that? Okay, first one. Oh, you hold on.
Can you barely see it?
Yeah, well, let me get, I got it.
Smooth.
I bet that's smooth. Yeah.
Hold on, hold up. Yeah, see this. I think we're gonna need to look at it. Okay, first of all, does this appear to have striations? Possibly.
It looks straight.
Looks like.
That actually might be like. Because of the nuclei.
Right, sorry, here we go, in the dark. Now you see, you can see the striations, right? And you can see this, even though it's a little wavy here because of the way it was prepared, you can see the cells in parallel, and like you said, there are nuclei out on the periphery, there's more than 1. So, yeah, so you're right, this is skeletal muscle, that's the easy one. The next one, I wanna tell you that you
Everyone
can,
has candles.
what'd you say?
So it looks like everyone has candles.
Yeah, it does, oh, it does, you know what, it looks really cool in here. Oh, this is so romantic. Who knew that we were going to have, we discussed relationships in chemistry, and now we have a romantic candle with microscope. Who says physiology is not romantic? They don't know. Well, you know what, so if you go to the next one, I'm gonna see if you know what this one is. This one is a little bit trickier, and truthfully, to find it, you should really go to like the edge of the tissue preparation, because that is cardiac. How do you know it's cardiac?
Cause it just seems like it.
Great answer. Honest. Great answer. Okay. You can see these dark lines here, which are our intercalated discs. And you can also see some branching. And in between the intercalated disc is the cell. And you can see they're very short, one nucleus there, and so forth. But like I said, for this one, if you want to look at cardiac muscle tissue, you gotta look to the edge of the slide, because remember that in the center of the heart, you have the circular arrangement of the way that the muscle is, because you know the ventricles, how they contract, so it doesn't look quite like this. And then of course the last one, which I will find here.
We're just doing so well in this class.
Actually, you guys did do well. You identified it correctly, which was good. Right, and the last one. Very good. Yes, it is smooth. Because why? Ashton, what do you think? Why is it smooth muscle?
Why is it smooth muscle?
Yes. I thought right away you would say because it's the last one.
So when you slid on hard surfaces, it'd be easier to tear.
Okay,
Because it's in the organ.
usually you don't find it in places that you tear, hopefully. I mean, like the digestive system and the bronchioles and the blood vessels, usually where you find, you don't really. No, the cells here are like a little bit more spindle shaped and it's arranged in a sheet. I love it. Where'd the candles go? The candles went out.
I made a wish though.
Alright, y'all, don't hurt yourselves. Let me turn the light on.
You come look at this.
Everybody's like, I'm afraid you're gonna stampede and like fall down because you
Off the
can't
car,
see.
so you can.
Leave. Okay, yes.
Just to know if that's smooth.
Yep. Yes.
Make sure to take my pictures right now.
So stick our phone in there.
Yeah, it's one of the best places you can see it, like around blood vessels, like we saw in some of the other tissues we looked at. That's the good place to look and find it. Thanks, guys. I'll see you in the.